An EPFL scientist has pinpointed the signs of brain activity that make up our brain fingerprint, which is unique like our regular fingerprint. EPFL scientist Enrico Amico learned that every one of us has a brain fingerprint and that this fingerprint constantly changes in time. His findings have just been published in the journal Science Advances. He says that his research examines networks and connections within the brain and especially the links between the different areas in order to gain greater insight into how things work. His research group processes the MRI scans to generate graphs represented as colorful matrices that summarize a subject's brain activity. This type of modeling technique is known in scientific circles as network neuroscience or brain connectomics. All the needed information is in these graphs that are commonly known as functional brain connectomes. The connectome is a map of the neural network. They inform us about what subjects were doing during their MRI scan, if they were resting or performing some other task, for example. Our connectomes change based on what activity was being carried out and what parts of the brain were being used. A few years ago, neuroscientists at Yale University studying these connectomes found that every one of us has a unique brain fingerprint. Comparing the graphs generated from MRI scans of the same subjects taken a few days apart, they were able to correctly match up the two scans of a given subject nearly 95% of the time. In other words, they could accurately identify an individual based on their brain fingerprint. Enrico Amico decided to take this finding one step further. In previous studies, brain fingerprints were identified using MRI scans that lasted several minutes, but he wondered whether these prints could be identified after just a few seconds, or if there was a specific point in time when they appear, and if so, how long would that moment last? Until now, neuroscientists have identified brain fingerprints using two MRI scans taken over a fairly long period. But do the fingerprints actually appear after just 5 seconds, for example, or do they need longer? And what if fingerprints of different brain areas appeared at different moments in time? Nobody knew the answer, so Amico's team tested different time scales to see what would happen. His research group found that 7 seconds wasn't long enough to detect useful data, but that around 1 minute and 40 seconds was. His study also showed that the fastest brain fingerprints start to appear from the sensory areas of the brain, and particularly the areas related to eye movement, visual perception, and visual attention. As time goes by, also frontal cortex regions, the ones associated to more complex cognitive functions, start to reveal unique information to each of us. The next step will be to compare the brain fingerprints of healthy patients with those suffering from Alzheimer's disease. Enrico Amico says that based on his initial findings, it seems that the features that make a brain fingerprint unique steadily disappear as the disease progresses. It gets harder to identify people based on their connectomes. It is as if a person with Alzheimer's loses his or her brain identity. Along this line, potential applications might include early detection of neurological conditions where brain fingerprints disappear. Amico's technique can be used in patients affected by autism or stroke or even in subjects with drug addictions.